The overall goal of this experiment is to evaluate the intracellular location of reactive oxygen species within Solea Senegalensis spermatozoa by confocal microscopy.
Reactive oxygen species have been usually associated to defective sperm function, causing cellular damage at different levels. Spermatozoa could be particularly sensitive to oxidative stress, since lipid peroxidation can produce a negative effect on motility and fertilization. Reactive oxygen species can even trigger an intrinsic apoptotic cascade. Peroxide is released from the mitochondria and it's able to penetrate the nucleus, affecting the DNA because it's a small and chargeless molecule. In this protocol, we use a fluorophore that, after being incorporated into the cell, can be modified by oxidative reaction, emitting green fluorescence. Therefore, you can know exactly the localization of reactive oxygen species.
In this video, we work with Solea Senegalensis spermatozoa, but you can use this protocol with other fish species with commercial interest, or with models like zebrafish or medaca. Some examples of the use of this technique in fish reproductive biology or aquaculture fields are the evaluation of cryopreservation thawing protocols, the comparison of reactive oxygen species levels before and after fish sperm activation, or you can correlate reactive oxygen species levels with good and bad breeders.
First of all, prepare the stocks of your fluorochromes. Take into account that you must perform fluorochrome manipulation according to safety data sheet recommendations. Special care should be taken while weighing and preparing the stocks. Keep the stock solutions in aliquots at -20